Okay, so in this video we're going to be looking at urine physiology, and the first part is the filtration. So this guy here, Johann Pim Kolff, he was important because he made major discoveries in the field of dialysis. So we can thank him for a lot of the technology involved in dialysis of kidneys. These are the areas for this particular part, this particular unit you'd be interested in. We don't do the colouring book, so we'll ignore that. So we're going to look at glomerular filtration. Glomerular filtration is basically the filtering of the blood in the glomerulus inside the Bowman's capsule of the nephron. You'll remember that. So we have something called the juxtaglomerular apparatus. So that means, remember you have juxtamedullary nephron, which was near the medulla. This is the apparatus that's near the glomerulus. Juxtaglomerular apparatus, to do with the blood vessels and stuff. Area where the distal end of the ascending limb of the loop of Henle lies in direct contact with the afferent arteriole. The afferent arteriole is the one going in to the glomerulus. Okay. This juxtaglomerular apparatus helps to regulate your blood pressure and the rate of filtration of the blood in the kidneys. It's got two main components: the macula densa, which is a group of strange-looking epithelial cells that are tightly packed together, and the juxtaglomerular cells, which are smooth muscle cells in the wall of the afferent arteriole. And these make renin, which is an enzyme, and that's involved in blood pressure regulation, which we'll see later. Remember, the cortical nephrons are the ones that are higher up inside the cortex of the kidney, whereas the juxtamedullary ones are much further down and have this big long loop of Henle and the vasa recta next to it, or wrapped around it, I should say. So we produce approximately 180 litres of filtrate in our kidneys per day. Okay, so that is 90 2-litre Coke bottles of filtrate per person per day. Do we pee that much out? Do we drink that much? I hope the answer for all of you is no, because I don't think we do. Okay, so let's have a look. It's a passive process, filtration. Have a think about that. What happens to all this filtrate? It's a passive process. That means there's no direct energy involved. The blood pressure powers the filtration. Okay, and the blood vessels get narrower inside the glomerulus, and that increases the pressure in there. Unlike a normal capillary bed, where the pressure decreases, inside the glomerulus it actually increases and aids filtration. So it goes across the filtration membrane. So you've got to get the filtrate from being in the blood to being out of the blood, and it has to cross a membrane, and that membrane is called the filtration membrane. Okay. Key to everything is a very large surface area, just like everything else we've studied. And our glomerular filtration rate is how fast we produce filtrate, which is about 180 litres per day, or 380 pints, and 125, which is about 125 millilitres per minute. And here's the answer to the question. We absorb most of it. If we didn't, we would be peeing all the time and drinking all the time. And the 180 litres per day is approximately 60 to 65 times the volume of our blood, the whole volume of the blood. Just think about that. So here we have some apparatus here to show you. What's happening here is you have the cortical radiator artery. Remember that when we looked at the anatomy? Well, there's a branch of that called the afferent arteriole, and it and it takes the blood into the glomerulus here, where filtration occurs. So filtration is occurring at this point. So the pressure inside these capillaries is much higher than outside, and it forces much of the liquid part of the blood out, along with everything that's dissolved in it. It then exits through the efferent arteriole. And if you look, the afferent arteriole is much fatter than the efferent arteriole, because the efferent arteriole doesn't carry as much blood, because we've lost a lot of the fluid part here. However, what happens next is this juxtaglomerular apparatus means that this efferent blood vessel then lies next to the tube where all the filtrate has just gone in, and reabsorbs. We have tubular reabsorption, where we take in a lot of the stuff, and you saw on the previous anatomy slides how much of each component of the filtrate is reabsorbed at this point. And we also secrete some waste products. Again, it's in that chart that you can look at. We secrete some waste products here that we couldn't get rid of here. Okay, we actually use energy to get rid of them. So the renal corpuscles are very efficient, because the capillaries are very long and extensive, lots of surface area, and the blood pressure is high. Okay, and the endothelial capsular membrane is thin and permeable. So thin means obviously that there's not a great distance to get across on the filter, and permeable means it allows stuff through. And so blood will flow rapidly through the glomerulus as well. So the speed that it goes through the glomerulus, the pressure that it generates inside the glomerulus, and the permeability of the membrane can all be changed. And any changes of any of these factors here will affect the glomerular filtration rate, how much filtrate we produce in a given period of time. Obviously, if the blood flowed less rapidly, you get a lower GFR. If the endothelial membrane became thicker or less permeable, we'd reduce the GFR. If the blood pressure was reduced, we'd reduce the GFR. And if somehow the surface area was reduced, we'd reduce the GFR. So this is a close-up of part of a capillary in the glomerulus, and you can see these little pores. This is where stuff can come out, and the pores are covered in this this visceral component of the lining of the Bowman's capsule, and these little false feet, these little projections here, slit filters. They can cover the holes, and we can change the filtration rate there. And we can see that in a minute. These podocytes, I should say, with the filtration slits. So here's one you've seen before, the afferent arteriole coming in, the efferent leaving, that's smaller. And this is the glomerular capsular space, and this is the visceral layer of the glomerular capsule here. And close up, it looks like this. And we have filtration slits, and we have fenestrations under the filtration slits. And the fenestrations are the holes that let stuff out, and these podocytes and slits can be controlled to adjust the amount of filtrate that can be let through. So pressures that affect filtration. The glomerular blood hydrostatic pressure. In other words, the blood pressure. So the blood pressure, and the figure we're going to give to this, it's 50 millimeters of mercury. You're familiar that millimeters of mercury is used as a pressure. We did it in the lungs. Well, this is the pressure here is 60 pushing out. So inside the blood vessel, inside the capillary, pushing out through the filter is 60. But pushing back is 15. Because the capsular hydrostatic pressure, basically the filtrate in the capsule pushes back against the blood. And so that would make it 45 total. But we also have blood colloidal osmotic pressure, the pressure caused by osmosis, because that's how the blood gets out, the filtrate gets out, which is 27. So my calculation, that would be 3742. So you've got 42 pushing back. So your net pressure would be this minus this plus this, which will equal, I believe, in my calculations are correct, 18. So you have a net outward pressure of 18. You might be expected to do a calculation like that. So this is showing you even more close-up of the membrane. Here's the plasma. This is the capillary endothelium. Then we have a basement membrane, and then we have a foot process of the podocyte. So you can see these are the slits through which the fluid can come out. Okay, so this is another set of figures. The important thing that you know is that your 55 is pushing out, and here you've got 45 pushing 45 pushing back in, which means you've got a net pressure of 10 pushing out. Okay, but you may have to perform that calculation. As long as this number wins, you're producing urine. Oh, potentially urine. You're producing filtrate anyway. So how do we regulate our glomerular filtration rate? So it depends mostly or greatly on the amount of blood that goes into the glomerulus, as you could imagine. No blood going in, no filtrate. Lots of blood, lots of filtrate. And the blood flow into the glomerulus depends on your systemic blood pressure and the diameters of the afferent, the one going in, and the efferent, the one going out, arterioles. And they're regulated by 3 mechanisms. So just think about that. You've got the blood going in through the afferent arteriole, your blood coming out through the efferent arteriole, and your blood pressure. So you can adjust the blood pressure, or you can adjust the diameter of the afferent arteriole. If you narrow that down, you're going to let less in. If you widen it up, you're going to let more in. Dilate it. On the efferent one, if you dilate the efferent one, it will cause a back pressure into the glomerulus and more filtration will occur. If you widen the efferent one, dilate the efferent one, then it will allow the blood to move more freely away from the glomerulus and not through the filtration mechanism. So the 3 mechanisms that can control this: autoregulation of the kidney itself, hormonal regulation, obviously by hormones, and nervous control, neurocontrol. As you could probably have guessed the second two, and the first one is autoregulation. It does it itself. So how does it do it itself? Well, autoregulation of the glomerular filtration rate is a constantly active mechanism which the juxtaglomerular apparatus that we mentioned at the beginning and parts of the afferent arteriole work together to maintain this control over the blood flow, despite changes in the systemic blood pressure. So that's really like a mechanical buffer. If you are in a situation where your blood pressure's gone up because perhaps you're running or something like that, you may not need more blood flowing through your kidneys. You may not need more blood going through your glomerulus. You may not need to filter more blood. So you can have this autoregulation, which senses changes in the blood pressure and changes the afferent arteriole, its diameter, so that the amount of blood going into the glomerulus remains pretty constant. So if the blood pressure goes up, you can close down the afferent arteriole and reduce the amount of blood going into the glomerulus. If the blood pressure goes down, you can widen it and allow more blood in. It's not completely understood. That means we don't know how it works, but it's thought to detect excess filtrate in the renal tubule and secrete a chemical that triggers the narrowing of it. So that would make sense. If you monitor how much filtrate you're making and you're making too much, then you can have a feedback mechanism that will slow down the amount that you're making. And the afferent arteriole can adjust its own diameter compensating for changes in blood pressure as well. Okay. So hormonal regulation involves the activities of the hormone angiotensin II and the activities of the hormone atrial natriuretic peptide, AMP. Okay, so we'll see how they work. Angiotensin II. Sounds like a sequel. Well, there is an angiotensin I, which we'll meet in a second. So, the production of this hormone, angiotensin II, occurs in the following steps. Renin is secreted by the juxtaglomerular cells in that juxtaglomerular glomerular apparatus that we met at the beginning of this slideshow. And renin's an enzyme, and it goes into the blood and travels around in the blood. And then it converts a plasma protein in the blood called angiotensinogen. Remember, the ogen is a precursor. Remember when we did all those enzymes in the gut, and they all ended in ogen because they didn't work? Well, this is the same. This produces, renin produces angiotensin I from angiotensin. So the liver makes the angiotensinogen, goes into the blood, and it meets the renin in the blood and becomes angiotensin I. Angiotensin I does not do the job we want. But it circulates to the lungs, where it encounters something called angiotensin converting enzyme, ACE. Now, those of you in the medical field might think, oh, ACE inhibitor, I've heard ACE, and you would be right, and we'll come to that shortly. The angiotensin converting enzyme converts angiotensin I to angiotensin II, which is the active hormone. Okay? So, angiotensin II does the following, and it's somewhat counterintuitive. Once it's formed, it does several things. It promotes vasoconstriction of the afferent arteriole. So that's the efferent arteriole. The efferent arteriole is the one that's leaving the glomerulus. And remember, we said if you constrict that, you close it up, cause a backlog of blood or a backup of blood in the glomerulus, increasing the pressure in the glomerulus, and therefore increasing the amount of filtrate being produced. Okay. It also stimulates the adrenal cortex of the adrenal gland and makes that secrete aldosterone, which is another hormone. And the aldosterone increases the reabsorption of water and sodium from the filtrate in the tubule. And this will in turn increase the blood volume and the blood pressure. Okay, let's do C and then I'll talk through them. It stimulates the posterior pituitary gland in the brain to secrete antidiuretic hormone, or vasopressin. And that causes an increase in reabsorption of water from the filtrate in the renal tubule, and that will increase the blood pressure. And it stimulates the thirst center in the hypothalamus to make you drink. Now, why does it do all these things? If you are losing fluid, too much fluid, your blood pressure will fall, you'll be thirsty, you'll be dehydrated, your osmotic balance will be upset. So we have a mechanism for controlling the amount of fluid in the blood. Okay. Now, blood pressure, one of the things about blood pressure is the more blood you have, the higher your blood pressure. And you can change the volume of the blood by removing water from it. So if your blood pressure begins to fall, this mechanism here tries to reabsorb as much water as is possible from the filtrate that you're making, and also makes you thirsty. So you go and drink some more water. Okay? So let's have a look at the first one. Promotes vasoconstriction of the efferent arteriole. So that makes you make more filtrate, which is a bit weird. It makes you make more filtrate in order so you can absorb more water. Okay. The second thing it does is stimulates the adrenal cortex and this aldosterone, which causes you to reabsorb water from the renal tubule. You're keeping your water. You're making urine more concentrated. It stimulates ADH to make you reabsorb more water. So you're taking as much water out of the filtrate as possible. So you're making a very yellow, concentrated urine because you're keeping the water. And the last thing it does, it says, hey, I want you to drink because you need to get more fluid in your blood and get your blood pressure up. So I'm going to make you feel thirsty. Now, going back to the previous slide, which is important here, is we're talking about this ACE, angiotensin converting enzyme. If you have high blood pressure, one of the standard pills that they will put you on, and I was on it for a while. I'm not on anything now, but I was on an ACE inhibitor for a while, and an ACE inhibitor inhibits ACE, the angiotensin converting enzyme. So it inhibits the production of angiotensin 2. So it inhibits all these things here so that you don't take in more fluids. So your blood pressure stays lower. Okay? Get the idea? If you're not producing as much angiotensin 2, then you're not reabsorbing water from anywhere, and you're not as thirsty as you would be. All right, that's good stuff, isn't it? So the next one is atrial natriuretic peptide AMP, and that's secreted in the atria of the heart when the heart stretches. So when the heart is stretching too much, it produces this atrial natriuretic peptide. Okay, and this increases the permeability of the endothelial cells in the capsular membrane. In other words, it makes your glomerulus more permeable to water. It lets more fluid out, and that will basically reduce your blood pressure, because the heart is, if there's too much blood, the heart strains. So this was a method of releasing that pressure. And finally, you've got the sympathetic division of the ANS, which can constrict the afferent arteriole when the blood pressure is was too high for the kidneys. Okay, stops fluid going in there. All right, and the result is temporarily reduced GFR. Okay, so what's filtered, reabsorbed, and/or secreted? The filtration is that the substance filtered into the Bowman's capsule include all these. This is just the same as in the last slideshow you saw, and it then goes on about what's factors influencing this filtration rate, explaining the role of renin-angiotensin. Okay, well, factors involving the regulation of sodium are surface area available for filtration, obviously, the filtration membrane permeability, which we mentioned before, how easily stuff gets across it, the pressure of the blood inside the glomerulus. The higher the pressure, the more filtration takes place. Okay, blood hydrostatic pressure is the most important factor influencing the glomerular filtration rate and potassium levels. Okay, we have intrinsic controls of the filtration rate within the kidney and extrinsic from outside. So you know we discussed those. Intrinsic is its self-control. Filtrate compared with plasma. Everything comes from plasma. Glomerular filtrate, similar to plasma, except in healthy people there's virtually no protein. So state the location and percent of reabsorption of glucose, amino acids, water, and ions. So we see that. That's where it's all being reabsorbed. So you need to know that glucose, amino acids, etc., most of the water, most of the sodium, most of the bicarbonate, most of the chlorine, most of the potassium are reabsorbed in the proximal convoluted tubule. We will spend quite a bit of time looking at the loop of Henle, but most reabsorption is the proximal convoluted tubule, and don't forget that. Okay. In people with uncontrolled diabetes, not all the glucose is reabsorbed, and ketone bodies can be detected in the urine. So you can detect glucose in the urine. Is the urine of people who are secreting glucose tastes sweet. And there was when I was a young man, a friend of mine was learning to be a doctor, and he said that the professor had passed round this flask of urine, and he got the urine and he dipped his finger in it and tasted it, and he said, "You'll see it's sweet." And he passed it round everybody in the room, all the students, and they wouldn't want to do it, but he was a professor, and he'd do what the professor says. And he goes all the way around the room, comes back to him, and he says, "You would have noticed that I dipped this finger into the urine and licked this finger." So he didn't ever taste it, but all his students did. So I think this is, we're going to talk about reabsorption in the next slideshow. Thank you.